I want to start with my story, that how I ended up with this talk. So actually, I started. I got a new position in the Yonsei University. Actually this week, and I was asked that, okay, you're welcome to Yonsei University, and you give a talk about genomic data analysis. Okay, I couldn't resist. Actually, I have a couple of papers on it. So actually, so my group is working on the quantum algorithm development, and then generally in this community have an attitude like, okay, here's a QUBO problem. Let's apply it, right? It's a genomic problem. Okay, we write a paper on it, right? So here's a problem in portfolio optimization. Okay, let's write a paper on it, right? So without understanding that much about it. So I'm one of them. So please understand that my lack of knowledge on the genomic analysis, but I will try my best. Okay, why do we have, okay, including me, have that kind of the attitude? It's because quantum computing is arguably good at optimization. So I said that the arguably means so we need to set the problem in a proper way, and then we need to utilize all the structures of the problem in a proper way to get the speed up. I listed some quantum algorithms for the quantum optimization solvers with some different features in it. Right? So today I will mostly talk about the quantum annealer and then combinatorial optimizations, and then we have the annealers, and then we have commercially available the machines, D-Abe and Pascal, and QA-OA is quite popular in combinatorial optimizations. And we can also use other optimization the algorithms in finding ground state. So we can map the combinatorial optimization problem in finding the minimum of the energy. Okay. Then I think as in a previous talk, it's already been explained that mostly this combinatorial optimization problems are NP hard, and then we ask the question that can we really expect a quantum advantage in NP hard problems? And as already been explained, that we don't expect that the worst case scenario, but we expect some average hardness, average case, right? So we can exploit some internal structure of the problem, and then we expect some biological problem have some structures, right? But it is very difficult to prove that it has some clear quantum advantages. So far in the quantum community, that we were quite happy with the sampling problems, quantum sampling, like a boson sampling, Gaussian boson sampling, or random circuit sampling. It's been quite successful because we washed out all the structures of the problems. Right? Yeah, but we know that quantum computer cannot solve the NP hard problem, but we expect that we can do something for some biological problems. Right? Okay. Here I want to add a couple of algorithms that my group is working on. Just to briefly introduce what kind of tools that we have. So one is a finite. So finite is we use the imaginary time propagator, imaginary time evolution, as ITE is called, and then this operator is a non-unitary operator, and then we implement it in the quantum circuit with the LCU. It's a linear combination of unitaries. And then we amplify the amplitude with the finite, the fixed point quantum amplitude, so developed by the Grover. So we try to solve some NP-hard problem, the max cut problems. And this problem also, of course, can be mapped into other problems. And then we also can apply to not only the QUBO, but HUBO problems. And yeah, so these kind of circuits are used to solve the problems. And then there was a problem why we don't use just ITE directly, because it's using ITE, and then as it approaches to the infinite, so 0 Kelvin, when it approaches to the 0 Kelvin, then it should end up with the ground state. But depending on the graph, we found out that some amplitude vanishes. So that's why we try to stop at some finite temperature, and then we optimize the problem, and then we amplify the amplitude, and then we obtain some fidelity amplifications. Okay. So the paper is on the way. So please wait for the paper, and then the click problem. And then we have a click problem solver. So click problem is we have some complex graph, and then we find some subgraph having some complex complete graph. That's a click problem. So we can approach that problem by introducing decay state. So decay state is the equal superposition of the n qubit states with a Hamming weight, a fixed Hamming weight k. So for example, so D42, so 4 choose 2. So we have a 4 qubit system, and then we have all the patterns can be generated. So we have a 6, and then this state is produced. Then we confine the Hilbert space within this subspace. Then we make some optimization problems to find the click. So by the way, so we try to use this this circuit to we try to apply this circuit to this protein interaction network, and it was introduced by Professor Sergi in the previous talk. Sorry, it was Professor Han, right? Professor Han. Okay. Okay. Then I want to also comment on the initial state preparation. Let's say that we have a, so for example, when we use the quantum annular, and then our initial state is always the uniform state. So uniform, it was prepared in a uniform space, right, with the n qubit, and then there's a 2 to the n states are superposed. Then what happens is that in searching problems, actually the quantum optimization is a searching problem, and in the searching problem, that's normally we are interested in the ground state, and then ground state's probability is very important. So actually it affects the efficiency of the quantum algorithm. So for example, let's say that we have this is the energy distribution of the initial state. So when we have the mean energy here, right? So we can expect that when we run the quantum algorithm, then we expect to have the lower energy states with high probabilities. Right. So yeah, and then if we have the some classical algorithm that some end up with this state, then we don't expect the much gain from that problems. Yeah. So what we expect is that if we can obtain some energy distribution profile of the quantum state, or we can engineer the quantum state for the quantum optimization problems, then we can gain much quantum advantages in optimization problems. So actually, we found a way to produce this kind of profile by computing the moments of the Hamiltonian. Then we can produce this kind of profile. Then we can judge whether that quantum state is good or not. And we expect that for example, quantum in the quantum circuit, we can implement arbitrary the initial state that we can design. But in the quantum annealer, that function is not provided. But I expect that the quantum annealers can use the user design the initial state. Then that would be really nice. Okay, now we know that we have solvers. Then we have solvers for the combinatorial optimizations problems. And then I want to say that many problems in bioscience can be mapped into combinatorial optimization. So we have some biological problem, and then we convert it into the combinatorial problems, and then we transform it into the energy problem, energy optimization problem, and then quantum computing generates a solution, and then we interpret the solution biologically. So we do it in that way. So my group applied these approaches to 3 topics. One is a codon optimization, and next is a multiple sequence alignment. It was introduced in the last talk, and then molecular docking. Okay, so codon optimization. Okay, so codon optimization is a very, very interesting problem. Like, the protein is consisted of the amino acids, and then single amino acid is encoded with the 3 letters of the nucleotides of the RNA. So ATUC, yeah, ATUC. Okay, so then the problem is each amino acid can have different codons. That the 3 letter is called codon, and then these codons can have so many codons can map into the single amino acid. That means if we have the a long amino acid sequence, like a protein, then we have really exponentially many number of sequence of the nucleotides. Then this is really amazing that I say that this might be some kind of error correcting procedure, so it can prevent some errors, and also it does some the resource saving. So some different species, different organisms uses different codons, and then it should adapt to the different environment. So different environment have different density of different nucleotides. Then it has to process this translation process with the ribosome to synthesize the protein, and then with a different environment. So that's a codon optimization. So different species use different codons because they have a different environment. So for example, if we want to produce insulin, and a human codon, and then the E. coli codon, so we produce the insulin with the E. coli, and then E. coli use different codon. So the mathematical problem occurs, so I would say the combinatorial problem occurs depending on the codon usage. So the codon usage is very interesting problems. That's it by utilizing it, so it maximizes the protein expression, and it improves the translation speed and accuracy, and then it enhances the protein folding. So when it translates, so when it catches the amino acid, the ribosome, then it's the different codon generate a different speed, different rate. So it will affect that how the protein folds. So it is very important to properly express the protein with the codons. Then, of course, then it will affect the stability of the RNA structures. In codon optimization model, we use the four the parameters. So codon usage bias, codon pair usage bias, then and the GC contents, and then we use all these parameters, and then we set some Cubo problems, and then we optimize it with a D-Wave machine. So it can be used by any machines. They can solve the Cubo problems. For example, QA/OA can solve the same problems, but we use a D-Wave because it can generate the moderate size of the solutions, and then the current circuit model is quite limited, so we decide to use D-Wave system. The output is it's an optimization problems. So this is a human host, then this is the goal, and then this is our result, and then the solution just sits in the bound. Okay. Then here is some challenges. Like we set the Cubo problems, and then that's it actually. Like we obtained some parameters from some journals from of the some biostatistics, and then there's not many things that we can do. Right? So we set a Cubo problem, then solver generates some answer. Then we can do some loop, but in the loop we expect that actually the real experiment. Right? So real experiment is needed actually to verify these results. Okay. In the codon optimization problems, we can expect a personalized some medicines, and then gene editing in the CRISPR, and some mRNA vaccine problems, and it involves. And then here's one problem has to be answered by our community, so including quantum and then the bioscience. Does a long-range correlation affect the biological function? So what does it mean? So in the classical algorithm, these codon sequences are sliced into the small chunks, and then they are optimized locally, and they are combined. In quantum, we expect that it is optimized in a full sequence. So that means its interaction range is long. It's very long, right? So the classical machine cannot access that kind of optimizations, right? But so far, people just use the classical algorithm, and then they use that that result for their biological experiments. And then we claim that long-range correlation would be important. Does biology use this kind of knowledge or not? So that's a kind of open question. Actually, we submitted the paper to the Cell sister journal, and it was actually rejected at the desk. And then we argued with the editor, and then I exactly brought this argument that it has a long-range correlation. It is amazing. Then the reaction was like, then bring the evidence. Right? Yeah. So we expect to have some evidence that this kind of effect would do something really nice. Okay. The multiple sequence alignment. It was introduced already in a previous speaker. So our approach is a little bit different. Okay, in the MSA problem, so actually MSA is done by pairwise comparison. So there are multiple sequences, and then classically it is important to find the proper sequence of the pairings. So we actually solved that problem. Okay, so we found the pairing sequences. It is called the guide trees, and this guide tree can be found with the traveling salesman's problem, which can be mapped into the QUBO. So we mapped it, and then we found the sequence alignments with these just classical algorithms. So we didn't really map everything into the quantum, but we found a guiding tree with the quantum machine. Okay. Yeah, so actually this part that we are expecting to have, but Professor Sergei already did so we want to map, we want to exploit some state fidelity to align the quantum, the multiple sequence alignment. But that's a really nice way to explore. Okay, so molecular docking. So molecular docking is a problem of the small molecule and then the aligning small molecule to the protein. Or there's a problem like a protein-protein docking. So protein-protein is how they are aligned. It's a very challenging mathematical problem. So if we consider just map it into some grid, and then the grid is evaluated with the occupation numbers, and then that evaluation is mapped into some kind of the energy functions. Then we optimize that energy. Then we got some patterns, some sequences, then which grids are occupied or not. There's a combinatorial optimization problems, and then there are several approaches that has been done, like a QUBO and then QA/OA, and some even the Gaussian Boson sampling use that. But all these approaches we are not so satisfied because it's quite example-oriented approach. Like that they generate solutions for some specific examples, but it does not work in general. So we have to improve it. We have to improve the method to generate some general problems. The limit is the hardware limit. We of course we have a hardware limit, so it's naturally gives us some limitations in the resolution of the energy grid, and also then there's some main problem is a lack of mathematical proof between docking score and the Hamiltonian energy. We just use some available the energy scoring functions, but we are not sure that if they are really good in describing this kind of problems. Yeah, so we need to think about how we map some biologically meaningful problem to the combinatorial optimization problem or physical problems. That's a really the question that we have to answer. Okay, here's a conclusion then. We have the quantum combinatorial optimization solvers, including the initial state preparation protocol. And then quantum tools are improving rapidly. But we need to find biologically important applications. This is very important part, and we need to find the proper models and proper analyses are essential. Right? These are really the important part. These are really lacking part in our community. So me, myself, as a quantum algorithm developer, we need to learn what would be the important biological problems. And bioscientists, I think bioscientists needs to learn then what kind of tools are available for their problems. So we need to talk actively to solve this really the important problem in our world. Thank you very much.